This is Father Faraz, and I will be presenting our work titled A Study of Biologically Plausible Neural Network, the Role and Interactions of Brain-Inspired Mechanisms in Continual Learning. This is a joint work with Bahram Zanuz and Elahi Irani. While the human brain excels at continually learning from a dynamically changing environment, standard artificial neural networks are inherently designed for training from stationary IID data. The sequential learning of tasks involved in continual learning violates this strong assumption, resulting in catastrophic forgetting of previously learned tasks. Now, although artificial neural networks are inspired by biological neurons, they omit numerous details of the design principles and learning mechanisms in the brain. We believe that these fundamental differences may account for the mismatch in performance and behavior of artificial neural networks and their biological counterparts. There are several differences. Biological neural networks are characterized by considerably more complex synapses and dynamic context-dependent processing of information. In addition, individual neurons have a specific role. Each presynaptic neuron has an exclusively excitatory or inhibitory impact on its postsynaptic partner, as postulated by Dale's principle. Furthermore, the distal dendritic segments in pyramidal neurons, which comprise the majority of excitatory cells in neocortex, receive additional context information and enable context-dependent processing of information. This, in conjunction with inhibition, allows the network to learn task-specific patterns and avoid catastrophic forgetting. We hypothesize that the context-dependent processing of information in pyramidal neurons, together with inhibition, may allow the network to learn task-specific patterns and avoid catastrophic forgetting. Here, we look at some of the biological mechanisms that are believed to enable efficient learning in the brain. Firstly, the replay of non-overlapping and sparse neural activities of previous experiences in the neocortex and hippocampus is considered to play a critical role in memory formation, consolidation, and retrieval. Furthermore, to protect information from erasure, the brain employs synaptic consolidation, in which plasticity rates are selectively reduced in proportion to strengthened synapses. Furthermore, it is also critical to consider how information is encoded in the brain. Neocortical circuits are characterized by high levels of sparsity in neural activations, and there is evidence suggesting that neuronal coding of natural sensory stimuli must be sparse. The goal of our work is to study the role and interactions of different mechanisms inspired by the brain in a biologically plausible framework for continual learning setup. To this end, we propose a more biologically plausible framework. Each layer consists of separate populations of exclusively excitatory pyramidal cells and inhibitory neurons, which adhere to Braille's principle. The shade in the figure indicates the strength of weights or activations, with the darkest shade indicating a higher value. As we can see, the pyramidal cells are augmented with dendritic segments, which receive an additional context signal, and the dendritic segment whose weights are more aligned with the context vector in the bottom row is selected to modulate the output activity of the feedforward neuron for context-dependent processing of information. Furthermore, the Hebbian learning step further strengthens the association between context and the winning dendritic segment with maximum absolute value, which is indicated with the darkest shade in the bottom row. Finally, heterogeneous dropout keeps the activation count of each pyramidal cell and drops the neurons that were most active for the previous task to enforce non-overlapping representations. The top-k remaining cells then project to the next layer. This provides us with a more biologically plausible framework within which we can study the role of different brain-inspired mechanisms and provide insights for designing new continual learning methods. We first study whether feed-forward neurons with separate populations of excitatory and inhibitory units can work well in the continual learning setting. Importantly, we note that when learning a sequence of tasks with inhibitory neurons, it is beneficial to take into account the disparities in the degree to which updates to different parameters affect the layer's output distribution. and hence forgetting. Our results show that models with feedforward neurons adhering to Dale's principle perform on par with standard neurons and can also further mitigate forgetting in conjunction with active dendrites when the quality of the context signal is high, as we can see in the permuted MNIST scenario. Another major difference is in the activation patterns. Neocortical circuits are characterized by high levels of sparsity in neural activations, which is in stark contrast to the dense and highly entangled connectivity in standard artificial neural networks. Particularly for continual learning, sparsity can provide several advantages. Sparse non-overlapping representations can reduce interference between tasks and can lead to the natural emergence of task-specific modules. We study the effect of different levels of activation sparsity by varying the ratio of active neurons in K winner-take-all activation function. Our results show that sparsity plays a critical role in enabling continual learning in deep neural networks, and as the sparsity and activations can effectively reduce interference by reducing the overlap in representations. And when we look at different scenarios, depending upon the similarity of subsequent tasks, we see that sparsity levels or the level of overlap that we have can have a significant impact in the results. Furthermore, information in the brain is encoded by the strong activation of a relatively small set of neurons that form a sparse coding. A different subset of neurons is utilized to represent different types of stimuli. There is also evidence of non-overlapping representation in the brain. To mimic this, we employ heterogeneous dropout, which, in conjunction with context-dependent processing of information, can effectively reduce the overlap of representations, leading to less interference between tasks and thereby less forgetting. Heterogeneous dropout can facilitate the formation of task-specific subnetworks and control the overlap and representation of different tasks. It provides us with a simple mechanism for balancing the reusability and interference of features depending upon the similarity of tasks, which can be quite beneficial when dealing with challenging continual learning scenarios. Additionally, for biologically plausible artificial neural networks, we believe that it is not only important to incorporate the design elements of biological neurons, but also the learning mechanisms it employs. Lifetime plasticity in the brain generally follows the Hebbian principle, which states that a neuron that consistently contributes to the firing of another neuron will build a stronger connection to that neuron. We follow the approach of complementing error-based learning with Hebbian update to strengthen the connection between contextual information and dendritic segments. The Hebbian update step increases the magnitude of the modulating signal from the dendrites on feedforward activity, which can further strengthen context-dependent gating and facilitate the formation of task-specific subnetworks. The brain employs different mechanisms to protect information from erasure. New spines are formed on different sets of dendritic branches in response to learning different tasks, which are protected from being eliminated through the mediation of synaptic plasticity and structural changes that persist when learning a new task. We employ synaptic consolidation by incorporating synaptic intelligence, which maintains an importance estimate of each synapse in an online manner during training and subsequently reduces the plasticity of synapses, which are considered important for learned tasks. In particular, we adjust the importance estimates to account for the disparity in the degree to which updates to different parameters affect the output of the layer. Our results show that employing synaptic intelligence in this manner further mitigates forgetting. Particularly for rotated MNIST with 20 tasks, it provides a considerable improvement in the performance. Now, another salient feature of the brain is experience replay. The replay of past neural activation patterns in the brain is considered to play a critical role in memory formation, consolidation, and retrieval. In particular, the failure of the model to avoid forgetting in the challenging class incremental learning setting without experience replay suggests that context-dependent processing of information alone does not suffice, and experience replay might be essential. Additionally, adding consistency regularization further improves performance as the model receives additional relational information about the structural similarity of classes, which facilitates the consolidation of information. Now, having shown the individual effect of each of the brain-inspired component in our biologically plausible framework, here we look at their combined effect. The different components complement each other and consistently improve the performance of the model. Hence, we achieve the highest performance gain when all of these components are combined together to form bio-ANN. Our key takeaway message, therefore, is that similar to the brain, employing multiple complementary mechanisms in a biologically plausible architecture is an effective approach to enable continual learning in artificial neural networks. Overall, our study presents a compelling case for incorporating the design principles and learning machinery of the brain into artificial neural networks and provides further credence to the argument that distilling the details of the learning machinery of the brain can bring us closer to human intelligence. Thank you.